Introduction. How important are amino acids? Consider sickle cell disease. People with sickle cell disease have red blood cells that, under certain conditions, can become rigid and sickle-shaped. Those sickle-shaped cells can become stuck in capillaries, blocking them. In severe cases, it can block enough of the blood supply to cause damage to several organs, such as the kidneys, liver, and spleen. This happens because of a mutation in hemoglobin, the protein in red blood cells that transports oxygen. Remarkably, the difference between the normal form of hemoglobin, HbA, and the one that causes sickle cell disease, HbS, is a seemingly minor one. All it takes is a change in a single amino acid on the surface of hemoglobin. The sixth amino acid in 2 of its 4 chains is changed from glutamic acid to valine. That minor difference allows the deoxygenated form of HbS to aggregate and precipitate, which leads to the sickled shape and all the symptoms of sickle cell disease. In this chapter, we'll take a look at the basics of proteins by focusing on the amino acids that compose them and how those amino acids contribute to the physical and chemical properties of proteins. 1.1 Amino acids found in proteins. Amino acids are molecules that contain 2 functional groups, an amino group, NH, and a carboxyl group, COOH. In this chapter, we'll focus specifically on the A amino acids, in which the amino group and the carboxyl group are bonded to the same carbon. The A carbon of the carboxylic acid. Think of the A carbon as the central carbon of the amino acid. In addition to the amino and carboxyl groups, the A carbon has 2 other groups which is attached to it, a hydrogen atom and a side chain, also called an R group, specific to each amino acid. The side chains determine the properties of amino acids, and therefore their functions. A note on terminology. Amino acids don't need to have both the amino and carboxyl groups bonded to the same carbon. For example, the neurotransmitter gamma-aminobutyric acid, GABA, has the amino group on the gamma carbon, 3 carbons away from the carboxyl group. Similarly, not every amino acid found in the human body is specified by a codon in the genetic code or incorporated into proteins. One example is ornithine, one of the intermediates in the urea cycle, the metabolic process by which the body excretes excess nitrogen. There are also some amino acids that are specifically modified for specialized roles in the body. For example, lysine is sometimes converted into pyrrolysine, and cysteine is sometimes converted into selenocysteine. That said, the Association of American Medical Colleges, AAMC, has specifically stated they'll focus on the 20 A amino acids encoded by the human genetic code, also called proteinogenic amino acids. So, for the rest of this chapter, we'll use the term amino acid to refer specifically to these compounds. Stereochemistry of amino acids. For most amino acids, the alpha carbon is a chiral, or stereogenic, center, as it has 4 different groups attached to it. Thus, most amino acids are optically active. The one exception is glycine, which has a hydrogen atom as its R group, making it a chiral. All chiral amino acids used in eukaryotes are L amino acids, so the amino group is drawn on the left in a Fischer projection. In the Kahn-Ingold-Prelog system, this translates to an S absolute configuration for almost all chiral amino acids. The only exception is cysteine, which, while still being an L amino acid, has an R absolute configuration because the CH2SH group has priority over the COOH group. Structures of the amino acids. There are several ways to classify amino acids. In this section, we'll break them down by the structures of their side chains. Nonpolar, non-aromatic side chains. 7 amino acids fall into this class. Glycine, discussed earlier, has a single hydrogen atom as its side chain and is therefore a chiral. It is also the smallest amino acid. Four other amino acids alanine, valine, leucine, and isoleucine have alkyl side chains containing 1 to 4 carbons. Methionine is one of only 2 amino acids that contains a sulfur atom in its side chain. Nevertheless, because the sulfur has a methyl group attached, it is considered relatively nonpolar. Finally, proline is unique in that it forms a cyclic amino acid. In all the other amino acids, the amino group is attached only to the A carbon. In proline, however, the amino nitrogen becomes a part of the side chain, forming a 5-membered ring. That ring places notable constraints on the flexibility of proline, which limits where it can appear in a protein and can have significant effects on proline's role in secondary structure. Aromatic side chains. 3 amino acids have uncharged aromatic side chains. The largest of these is tryptophan, which has a double ring system that contains a nitrogen atom. The smallest is phenylalanine, which has a benzyl side chain, a benzene ring plus a CH group. Adding an O group to phenylalanine gives the third member, tyrosine. While phenylalanine is relatively nonpolar, the O group makes tyrosine relatively polar. Polar side chains. 5 amino acids have side chains that are polar but not aromatic. Serine and threonine both have O groups in their side chains, which makes them highly polar and able to participate in hydrogen bonding. Asparagine and glutamine have amide side chains. Unlike the amino group common to all amino acids, the amide nitrogens do not gain or lose protons with changes in pH, they do not become charged. The last amino acid with a polar side chain is cysteine, which has a thiol, SH, group in its side chain. Because sulfur is larger and less electronegative than oxygen, the SH bond is weaker than the OH bond. This leaves the thiol group in cysteine prone to oxidation, a reaction we'll study later in this chapter. Negatively charged, acidic, side chains. Only 2 of the 20 amino acids have negative charges on their side chains' physiological pH, 7.4. Those 2 are aspartic acid, aspartate, which is related to asparagine, and glutamic acid, glutamate, which is related to glutamine. Unlike asparagine and glutamine, aspartate and glutamate have carboxylate, Cu, groups in their side chains, rather than amides. Note that aspartate is simply the deprotonated form of aspartic acid, and glutamate is the deprotonated form of glutamic acid. Positively charged, basic, side chains. The remaining 3 amino acids have side chains that have positively charged nitrogen atoms. Lysine has a terminal primary N group, while arginine has 3 nitrogen atoms in its side chain. The positive charge is delocalized over all 3 nitrogen atoms. The final amino acid, histidine, has an aromatic ring with 2 nitrogen atoms. This ring is called an imidazole. You might be wondering how histidine can acquire a positive charge. The pK of the side chain is relatively close to 7.4, it's about 6, so, at physiologic pH, one nitrogen atom is protonate and the other isn't. Under more acidic conditions, the second nitrogen atom can become protonate, giving the side chain a positive charge. Hydrophobic and hydrophilic amino acids. Classifying amino acid side chains as hydrophobic or hydrophilic is actually a very complex matter. For example, tyrosine has both an O group and an aromatic ring, so which one wins? A few clear conclusions can be drawn, though. First, the amino acids with Jong alkyl side chains alanine, isoleucine, leucine, valine, and phenylalanine are all strongly hydrophobic and thus more likely to be found in the interior of proteins away from water on the surface of the protein. Second, all the amino acids with charged side chains positively charged histidine, arginine, and lysine, plus negatively charged glutamate and aspartate are hydrophilic, as are the amides asparagine and glutamine. The remaining amino acids lie somewhere in the middle and are neither particularly hydrophilic nor particularly hydrophobic. Amino acid abbreviations. Now that we have explored the structures of the 20 proteinogenic amino acids, it is worth mentioning that you are expected to be able to identify an amino acid on the MCAT not only by name, but also by its 3-letter and 1-letter abbreviations. 1.2 Acid-base chemistry of amino acid. The AAMC loves to use amino acids to test your understanding of acid-base chemistry because they have both an acidic carboxylic acid group and a basic amino group. That makes them amphoteric species, as they can either accept a proton or donate a proton. How they react depends on the pH of their environment. The key to understanding the behavior of amino acids is to remember 2 facts. Ionizable groups tend to gain protons under acidic conditions and lose them under basic conditions. So, in general, at low pH, ionizable groups tend to be protonate; at high pH, they tend to be deprotonated. The pKa of a group is the pH at which, on average, half of the molecules that species are deprotonated, that is, protonate version of the ionizable group, deprotonated version of the ionizable group, or Ha equals A. pH is less than the pKa, a majority of the species will be protonate. If the pH is higher than the pKa, a majority of the species will be deprotonated. Protonation and deprotonation. Because all amino acids have at least 2 groups that can be deprotonated, they all have at least 2 pKa values. The first one, pKa, is the pKa for the carboxyl group and is usually around 2. For most amino acids, the second pKa value, pKa, is the pKa for the amino group, which is usually between 9 and 10. For amino acids with an ionizable side chain, there will be 3 pKa values, but we'll come back to that later. As an example, let's take glycine, which doesn't have an ionizable side chain. Positively charged under acidic conditions. At pH 1, below even the pH of the stomach, there are plenty of protons in solution. Because we're far below the pKa of the amino group will be fully protonate, NH, and thus positively charged. Because we're also below the pKa of the carboxylic acid group, it too will be fully protonate, COOH, and thus neutral. Therefore, at very acidic pH values, amino acids tend to be positively charged. Zwitterions at intermediate pH. If we increase the pH of the amino acid solution from pH 1 to pH 7.4, the normal pH of human blood, we've moved far above the pKa of the carboxylic acid group. Physiological pH, you will not find amino acids with the carboxylate group protonate, COOH, and the amino group unprotonated, NH. Under these conditions, the carboxyl group will be in its conjugate base form and be deprotonated, becoming Cu. Conversely, we're still well below the pK of the basic amino group, so it will remain fully protonate and in its conjugate acid form, NH. Thus we have a molecule that has both a positive charge and a negative charge, but overall, the molecule is electrically neutral. We call such molecules dipolar ions, or Zwitterions, from the German Zwitter, or hybrid. The 2 charges neutralize one another, and Zwitterions exist in water as internal salts. Negatively charged under basic conditions. Milk of magnesia, which is often used as an antacid, has a pH around 10.5. At that pH, the carboxylate group is already deprotonated and thus remains Cu. On the other hand, we are now well above the pKa for the amino group, so it deprotonates too, becoming NH. So, at highly basic pH, glycine is now negatively charged. Titration of amino acids. Because of these acid-base properties, amino acids are great candidates for titrations. We assume that the titration of each proton occurs as a distinct step, resembling that of a simple monoprotic acid. Thus, the titration curve looks like a combination of 2 monoprotic acid titration curves, or 3 curves, if the side chain is charged, shows the titration curve for glycine. After we inspect this curve, we'll look at the differences for the amino acids with charged side chains. Imagine an acidic I-molarity glycine solution. At low pH values, glycine exists predominantly as and NH2CH2CU. It is fully protonate, with a positive charge. As the solution is titrated with NaOH, the carboxyl group will deprotonate first because it is more acidic than the amino group. When 0.5 equivalents of base have been added to the solution, the concentrations of the fully protonate glycine and its Swiddle and NH2CH2CU are equal, that is, plus NH2CH2COOH equals plus NH2CH2CU. This point, the pH equals pK. Remember, when the pH is close to the pK value of a solute, the solution is acting as a buffer and the titration curve is relatively flat, as demonstrated in the blue boxes in the diagram. As we add more base, the carboxylate group goes from half deprotonated to fully deprotonated. The amino acid stops acting like a buffer, and pH starts to increase rapidly during this phase. When we've added 1.0 equivalent of base, glycine exists exclusively as the zwitterion form. Remember, we started with 1.0 equivalent of glycine. This means that every molecule is now electrically neutral, and thus the pH equals the isoelectric point, pi, of glycine. This is true of all amino acids. The isoelectric point is the pH at which the molecule is electrically neutral. For neutral amino acids, it can be calculated by averaging the two pKa values for the amino and carboxyl groups. For glycine, the pi value is 2.34 plus 9.60, divided by 2 equals 5.97. Remember that when the molecule is neutral, it is especially sensitive to pH changes, and the titration curve is nearly vertical. As we continue adding base, glycine passes through a second buffering phase as the amino group deprotonates. Again, the pH remains relatively constant. When 1.5 equivalents of base have been added, the concentration of the zwitterion form equals the concentration of the fully deprotonated form, that is, plus NH2CH2CU equals NH2CH2CU, and the pH equals pKa. Once again, the titration curve is nearly horizontal. Finally, when we've added 2.0 equivalents of base, the amino acid has become fully deprotonated, and all that remains is NH2CH2CU. Additional base will only increase the pH further. Amino acids with charged side chains. For amino acids with charged side chains, such as glutamic acid and lysine, the titration curve has an extra step, but works along the same principles described above. Because glutamic acid has two carboxyl groups and one amino group, its charge in its fully protonate state is still +1. It undergoes the first deprotonation, losing the proton from its main carboxyl group, just as glycine does. At that point, it is electrically neutral. When it loses its second proton, just as with glycine, it's over, all charge will be -1. However, the second proton that is removed in this case comes from the side chain carboxyl group, not the amino group. This is a relatively acidic group, with a pK of around 4.2. The result is that the pL of glutamic acid is much lower than that of glycine, around 3.2. The isoelectric point for an acidic amino acid can be calculated as follows. Lysine, on the other hand, has two amino groups and one carboxyl group. Thus, its charge in its fully protonate state is +2, not +1. Losing the carboxyl proton, which still happens around pH 2, brings the charge down to +1. Lysine does not become electrically neutral until it loses the proton from its main amino group, which happens around pH 9. It gets a negative charge when it loses the proton on the amino group in its side chain, which happens around pH 10.5. Thus, the ISO, electric point of lysine is the average of the pKa values for the amino acid. The pL is around 9.75. The isoelectric point for a basic amino acid can be calculated as follows. The take-home message: amino acids with acidic side chains have relatively low isoelectric points, while those with basic side chains have relatively high ones. Peptide bond formation in hydrolysis. Peptides are composed of amino acid subunits, sometimes called residues. Dipeptides consist of 2 amino acid residues, tripeptides have 3. The term oligopeptide is used for relatively small peptides, up to about 20 residues, while longer chains are called polypeptides. The residues in peptides are joined together through peptide bonds, a specialized form of an amide bond, that forms between the Cu group of one amino acid and the NH3+ group of another amino acid. This forms the functional group C(O)NH. In this section, we'll look at the key reactions involved in forming and breaking peptide bonds. Peptide bond formation. Peptide bond formation is an example of a condensation or dehydration reaction because it results in the removal of a water molecule, H2O. It can also be viewed as an acyl substitution reaction, which can occur with all carboxylic acid derivatives. When a peptide bond forms, the electrophilic carbonyl carbon on the first amino acid is attacked by the nucleophilic amino group on the second amino acid. After that attack, the hydroxyl group of the carboxylic acid is kicked off. The result is the formation of a peptide, amide, bond. Because amide groups have delocalizable pi electrons in the carbonyl and in the lone pair on the amino nitrogen, they can exhibit resonance, thus, the CN bond in the amide has partial double bond character. As a result, rotation of the protein backbone around its CN amide bonds is restricted, which makes the protein more rigid. Rotation around the remaining bonds in the backbone, however, is not restricted, as those remain single bonds. When a peptide bond forms, the free amino end is known as the amino terminus or N-terminus, while the free carboxyl end is the carboxy terminus or C-terminus. By convention, peptides are drawn with the N-terminus on the left and the C-terminus on the right. Similarly, they are read from N-terminus to C-terminus. Peptide bond hydrolysis. For enzymes to carry out their function, peptides need to be relatively stable in solution. Therefore, they don't normally fall apart on their own. On the other hand, in order to digest proteins, we need to break them down into their component amino acids. In organic chemistry, amides can be hydrolyzed using acid or base catalysis. In living organisms, however, hydrolysis is catalyzed by hydrolytic enzymes such as trypsin and chymotrypsin. Both are specific, in that they only cleave at specific points in the peptide chain. Trypsin cleaves at the carboxyl end of arginine and lysine, while chymotrypsin cleaves at the carboxyl end of phenylalanine, tryptophan, and tyrosine. While you don't need to know the exact mechanism of how these enzymes catalyze hydrolysis, you do need to understand the main idea. They break apart the amide bond by adding a hydrogen atom to the amide nitrogen and an O group to the carbonyl carbon. This is the reverse reaction shown here. 1.4 Primary and secondary protein structure. Proteins are polypeptides that range from just a few amino acids in length up to thousands. They serve many functions in biological systems, functioning as enzymes, hormones, membrane pores and receptors, and elements of cell structure. Proteins are the main actors in cells. The genetic code, after all, is simply a recipe making thousands of proteins. Proteins have 4 levels of structure: primary, 1 degree, secondary, 2 degrees, tertiary, 3 degrees, and quaternary, 4 degrees. In this section, we'll examine the first 2, we'll discuss tertiary and quaternary structure in the next section. Primary structure. The primary structure of a protein is the linear arrangement of amino acids coded in an organism's DNA. It's the sequence of amino acids, listed from the N-terminus, or amino end, to the C-terminus, or carboxyl end. So, for example, the first 10 amino acids of the beta chain of hemoglobin are normally valine, histidine, leucine, threonine, proline, glutamate, lysine, serine, and alanine. Primary structure is stabilized by the formation of covalent peptide bonds between adjacent amino acids. The primary structure alone encodes all the information needed for folding at all of the higher structural levels. The secondary, tertiary, and quaternary structures a protein adopts are the most energetically favorable arrangements of the primary structure in a given environment. The primary structure of a protein can be determined by a laboratory technique called sequencing. This is most easily done using the DNA that coded for that protein, although it can also be done from the protein itself. Secondary structure. A protein's secondary structure is the local structure of neighboring amino acids. Secondary structures are primarily the result of hydrogen bonding between nearby amino acids. The two most common secondary structures are helices and B-pleated sheets. The key to the stability of both structures is the formation of intramolecular hydrogen bonds between different residues. Alpha helices. The alpha helix is a rod-like structure in which the peptide chain coils clockwise around a central axis. The helix is stabilized by intramolecular hydrogen bonds between a carbonyl oxygen atom and an amide hydrogen atom 4 residues down the chain. The side chains of the amino acids in the alpha helical conformation point away from the helix core. The alpha helix is an important component in the structure of keratin, a fibrous structural protein found in human skin, hair, and fingernails. Beta-pleated sheets. In beta-pleated sheets, which can be parallel or antiparallel, the peptide chains lie alongside one another, forming rows or strands held together by intramolecular hydrogen bonds between carbonyl oxygen atoms on one chain and amide hydrogen atoms in an adjacent chain. To accommodate as many hydrogen bonds as possible, the beta-pleated sheets assume a pleated, or rippled, shape. The R groups of amino residues point above and below the plane of the beta-pleated sheet. Fibroin, the primary protein component of silk fibers, is composed of beta-pleated sheets. Secondary structures and proline. Because of its rigid cyclic structure, proline will introduce a kink in the pen chain when it is found in the middle of an alpha helix. Proline residues are thus rarely found in alpha helices, except in helices that cross the cell membrane. Similarly, rarely found in the middle of pleated sheets. On the other hand, proline is often found in the turns between the chains of a beta-pleated sheet, and it is often found as the residue at the start of an alpha helix. 1.5 Tertiary and quaternary protein structure. Proteins can be broadly divided into fibrous proteins, such as collagen, that have structures that resemble sheets or long strands, and globular proteins, such as myoglobin, that tend to be spherical, that is, like a globe. These are caused by tertiary and quaternary protein structures, both of which are the result of protein folding. Tertiary structure. A protein's tertiary structure is its three-dimensional shape. Tertiary structures are mostly determined by hydrophilic and hydrophobic interactions between R groups of amino acids. Hydrophobic residues prefer to be on the interior of proteins, which reduces their proximity to water. Hydrophilic NH and C equals 0 bonds found in the polypeptide chain get pulled in by these hydrophobic residues. These hydrophilic bonds can then form electrostatic interactions and hydrogen bonds that further stabilize the protein from the inside. As a result of these hydrophobic interactions, most the amino acids on the surface of proteins have hydrophilic, polar or charged, groups. Highly hydrophobic R groups, such as phenylalanine, are almost never found on the surface of a protein. The three-dimensional structure can also be determined by hydrogen bonding, as well as acid-base interactions between amino acids with charged R groups, creating salt bridges. A particularly important component of tertiary structure is the presence of disulfide bonds, the bonds that form when two cysteine molecules become oxidized to form cysteine. Disulfide bonds create loops in the protein chain. In addition, Disulfide bonds determine how wavy or curly human hair is. The more disulfide bonds, the curlier it is. Note that forming a disulfide bond requires the loss of 2 protons and 2 electrons, oxidation. The exact details of protein folding are beyond the scope of the MCAT, but the basic idea is that the secondary structures probably form first, and then hydrophobic interactions and hydrogen bonds cause the protein to collapse into its proper three-dimensional structure. Along the way, it adopts intermediate states known molten globules. Protein folding is an extremely rapid process. From start to finish, it typically takes much less than a second. If a protein loses its tertiary structure, a process commonly called denaturation, loses its function. Folding and the solvation layer. Why do hydrophobic residues tend to occupy the interior of a protein, while hydrophilic residues tend to accumulate on the exterior portions? The answer can be summed up in one word: entropy. Whenever a solute dissolves in a solvent, the nearby solvent molecules form a solvation layer around that solute. From an enthalpy standpoint, even hydrocarbons are more stable in aqueous solution than in organic ones, delta H. However, when a hydrophobic side chain, such as those in phenylalanine and leucine, is placed in aqueous solution, the water molecules in the solvation layer cannot form hydrogen bonds with the side chain. This forces the nearby water molecules to rearrange themselves into specific arrangements to maximize hydrogen bonding, which means a negative change in entropy, delta S. Remember that negative changes in entropy represent increasing order, decreasing disorder, and thus are unfavorable. This entropy change makes the overall process non-spontaneous, delta G0. On the other hand, putting hydrophilic residues such as serine or lysine on the exterior of the protein allows the nearby water molecules more latitude in their positioning, thus increasing their entropy, delta S0, and making the overall solvation process spontaneous. Thus, by moving hydrophobic residues away from water molecules and hydrophilic residues toward water molecules, a protein achieves maximum stability. Quaternary structure. All proteins have elements of primary, secondary, and tertiary structure. Not all proteins have quaternary structure. Quaternary structures only exist for proteins that contain more than one polypeptide chain. For these proteins, the quaternary structure is an aggregate of smaller globular peptides or subunits and represents the functional form of the protein. The classic examples of quaternary structure are hemoglobin and immunoglobulins. Hemoglobin consists of 4 distinct subunits, each of which can bind one molecule of oxygen. Similarly, immunoglobulin G, IgG, antibodies also contain a total of 4 subunits each. The formation of quaternary structures can serve several roles. First, they can be more stable, by further reducing the surface area of the protein complex. Second, they can reduce the amount of DNA needed to encode the protein complex. Third, they can bring catalytic sites close together, allowing intermediates from one reaction to be directly shuttled to a second reaction. Finally, and most important, they can induce cooperativity, or allosteric effects. We'll discuss this much further in the next chapter, especially for hemoglobin, but the basic idea is that one subunit can undergo conformational or structural changes, which either enhance or reduce the activity of the other subunits. Conjugated proteins. Conjugated proteins derive part of their function from covalently attached molecules called prosthetic groups. These prosthetic groups can be organic molecules, such as vitamins, or even metal ions, such as iron. Proteins with lipid, carbohydrate, and nucleic acid prosthetic groups are referred to as lipoproteins, glycoproteins, and nucleoproteins, respectively. These prosthetic groups have major roles in determining the function of their respective proteins. For example, each of hemoglobin's subunits, as well as myoglobin, contains a prosthetic group called heme. The heme group, which contains an iron atom in its core, binds to and carries oxygen. As such, hemoglobin is inactive without the heme group. These groups can also direct the protein to be delivered to a certain location, such as the cell membrane, nucleus, lysosome, or endoplasmic reticulum. 1.6 Denaturation. In the previous section, we discussed protein folding. The reverse of this process is denaturation, in which a protein loses its three-dimensional structure. Although it is sometimes reversible, denaturation is often irreversible. Whether its denaturation is reversible or not, unfolded proteins cannot catalyze reactions. The two main causes of denaturation are heat and solutes. As with all molecules, when the temperature of a protein increases, its average kinetic energy increases. When the temperature gets high enough, this extra energy can be enough to overcome the hydrophobic interactions that hold a protein together, causing the protein to unfold. This is what happens when egg whites are cooked. In the uncooked egg whites, albumin is folded, which makes it transparent. Cooking them causes the albumin molecules to denature and aggregate, forming a solid, rubbery white mass that will not revert to its transparent form. On the other hand, solutes such as urea denature proteins by directly interfering with the forces that hold the protein together. They can disrupt tertiary and quaternary structures by breaking disulfide bridges, reducing cysteine back to 2-cysteine residues. They can even overcome the hydrogen bonds and other side chain interactions that hold A helices and beta-pleated sheets intact. Similarly, detergents such as SDS, sodium dodecyl sulfate, also called sodium lauryl sulfate, can solubilize proteins, disrupting noncovalent bonds and promoting denaturation. Conclusion. Nearly every part of a cell involves proteins in some way, from the nucleus to the mitochondria to the cell membrane. The MCAT will test your understanding of key concepts regarding amino acids because the amino acids that compose a protein determine its structure. In the next chapter, we'll discuss the best known function of proteins, their role as enzymes.